We've got a great show today on reptile viruses with Dr. Shane Simpson and Dr. Joshua Llinas. As always, I'm your host, Yanni Torcola. My co-host for today, Ahlia Chanel, she's back. She's from UQ Environmental Science graduate.
Yep.
She's also a reptile keeper, and you used to do some reptile demo back in the day.
Yeah, I used to do reptile shows with Geckos Wildlife.
Geckos Wildlife.
Always a good time.
And you're still keeping a few
I'm still
animals?
keeping. Got a blue tongue, and I got a Darwin carpet, and soon to be a black-headed, and maybe another blue tongue.
Yeah, you might be getting Hamish
Yeah,
Noller's
I'm getting
black-headed
Hamish's
python.
black-headed. I'm pretty excited.
Yeah, it's
he
beautiful
agreed to give it to me
sound.
while drunk, and I held it to him.
Well, always take
She
people
wanted a
up.
poker game.
Yeah.
Always take drunken snake offers up.
Yeah.
It's the way to go. So look, in honor of our reptile show, we're drinking Snake Charmers. It's nectarine cordial, gin, contro, bitters, and saffron. We've also got a cheesecake with some whipped sour cream and some strawberries on top. We've also got a mocktail on deck.
Yes,
We've got
we
the
do.
blueberry mojito. Blueberry mocktail mojito. Excellent. And again, joining us as well, we've got two fantastic guests. One who you already know, Dr. Joshua Llinas, with his Bachelor of Science. Excellent to have you on. Always a joy. And with his Bachelor of Science from State Uni of New York in 2000. Is that correct? He then moved to UQ for a Bachelor of Vet Science and Masters in Vet Science at Murdoch Uni in Melbourne. So everybody goes down to Melbourne.
Over in Perth.
Perth, that's right. Oh, well,
Perth, yeah.
still down south.
Yeah.
Well, okay, so Murdoch Uni over in Perth, my bad. Currently the veterinary director at HerpVet, and they're at Jindalee Green Cross Vet Surgery. So HerpVet providing reptile and amphibian vet services here in Brisbane. You can check them out at herpvet.com.au or on Facebook.
Actually, tonight, website launches tonight.
Launches tonight?
Ooh.
Ooh, fantastic.
There's no black tie function?
Hopefully.
This is the black tie.
Oh, this is black tie.
Yeah,
Oh,
no,
okay.
it's his black
I
tie.
forgot it's pretty relaxed.
It's about as
I put on my good t-shirt.
Okay, yeah.
Black t-shirt? Yeah, I can put a tie on if I
I've
want
got,
to.
fancy
So-
sunnies, everything.
Yeah, it's fancy, very fancy. I'm not even wearing socks or shoes. So you've had some pretty cool Python cases come through. We
Yeah,
brought
it's
you
been
an
a
interesting one.
great couple of weeks.
Yeah, you've
you
been
brought
busy.
Me one. That was the tennis ball.
Tennis ball.
Yeah.
Python
came
who'd
in,
eaten a tennis
Oh,
ball.
I saw that.
Yeah, couldn't get it out manually.
Oh.
It just would not come out, so I went to surgery, and
Yeah.
it's doing pretty well. You've got it here now.
Yeah, so he's back here in recovery. He was actually found, I think, 2 streets down.
Oh,
So it's
excellent.
2 streets
That's
down
nice
from here.
and easy.
Yeah, nice and easy.
Nice
and
and
he'd eaten somebody's pet dog's tennis ball and then came hiding in the living room underneath their couch.
Better than one dropped off last night, which was the old man slipper.
But
He ate a whole slipper?
Yep.
A carpet python that ate a slipper.
Yep.
Wow.
So can you tell us that story real quick,
Shoot.
Josh? That was incredible.
Yeah. Oh, it just came in last night. Apparently went to the toilet, came back, and had eaten his slipper. It was very. You don't make this stuff up.
Yeah.
It was quite a good one. So that's being stabilized right now and going to surgery in the next day or two.
Yeah, wow. So what's how? I'm guessing a slipper is a little bit trickier than a tennis ball.
No, it should be just fine. Just go on the top. They're made to stretch, so everything comes out fairly
true.
well.
Well,
But
I mean, it might at least be a bit harder to palpitate a tennis.
it's actually quite a big snake, so you can only just see the bulge. The only reason I know is they sent me a photo of the matching one.
Imagine
I'm not aware of any published papers on slipper
so
extraction either.
That's
So.
right. We'll do that one.
Yeah.
You should write one.
To be presented at next
Yeah.
conference.
Yeah.
of course. Or on the
Absolutely.
you can come tell us about it on the show, of course.
We've had tennis balls, slippers, pillowcases. That's been in the last 2 months.
Yep.
Wow.
Black-headed pythons. They're like the Labradors of the snake
Oh, really?
world.
Yeah.
They'll
So
just
keep
eat
mine.
anything.
Just
Don't let mine
consuming
roam
everything.
around then.
Absolutely.
Wow.
They eat everything.
I've heard this
Insane.
one gets into weird places as well.
Yeah, they're
they're-
little explorers. I can tell
they're
you
big.
from having one
Yeah.
myself. They're pretty
Hamish is
roamy.
putting locks on my ankle.
All right, and moving on, we have our main guest for today, Dr. Shane Simpson, originally from Melbourne, moved up here to UQ to do
Yep.
your Bachelor of Science with honours in Bachelor of Vet
Vet
Science.
science, correct, yep.
Awesome. So graduated in 1996 and then back down to join the Carringal Vet Hospital in Frankston in the outer
Correct.
suburbs of Melbourne. So you're now a partner and practice owner there as well?
Yep, just ticked over 22 years there
Wow.
now. So first job after graduation and haven't left.
And straight into working with reptiles when you were there, or
No, not really.
slowly?
I started as a new grad. When I actually graduated, I wanted to be a specialist veterinary radiologist. So I wanted to do ultrasound and X-rays.
Okay.
And had an interest in the reptiles, but
Yep.
that came a little bit later. But
Yeah.
I was going to do ultrasound was my thing, and still is. I still do a lot of ultrasound work. So.
Well, yeah, very important in vet work, I imagine. Yeah, cool. Yeah. So also known as the Reptile Doctor Online, with a lifelong interest in reptiles and amphibians. Also, obviously, much of your vet focus is on reptile and amphibian medicine and surgery. You can see them at thereptiledoctor.com.au and on Facebook. Now, Dr. Simpson is around doing some talks. Reptile viruses, what Australian keepers need to know. He's up at UQ this afternoon, so it's going to be a bit too late by the time the show comes out to join the show. Is there any more talks on the tour plan?
Well, it's been pretty popular, this little talk show tour that I'm doing. So yeah, we did a couple of talks during the week down in Melbourne. We've had quite a good turnout, and I've sort of asked the audience, does this work for them? And so we might look at making it a bit of an annual roadshow event.
Just to keep
it's changing every year, so
Well, certainly the reptile from the viral point of view is, but I've sort of thrown out ideas of doing talks on things like reptile mites and metabolic bone disease and a lot of the common conditions that we see. And so it seems to be that people want to do it. So whether we continue on the same format as we are at the moment, but this is, to be honest, it was sort of a one-off sort of thing, in that. And with the RepX Expo, I sort of will tack onto that and see what sort of numbers we can get. And it's been really popular. So.
Yeah, fantastic.
I'd go.
Yeah, I would love to make it if we weren't super busy tonight already.
You'll be able to buy the ebook and the streaming video next week.
Oh, wonderful. Well, I may have to try to swing by tonight anyway if I get some time off out of the house. Now, look, the idea of doing more of these talks, I think that's awesome. Obviously, the virology field is pretty rapidly changing as well.
Yep.
So,
well, an example, I've literally got stuff in my talk tonight that was like yesterday. Kid you not. I actually spoke to a researcher yesterday, and we've tweaked a few things. So that's how up to date it is. It's that changing that quickly.
Well, then, no reason to not keep doing that.
So I'll be back next week doing a whole different talk.
Well, I mean, that's still interesting. You'll still obviously.
I'm not free to drive you next
week. Okay.
Well, then we have to cancel.
It is good, really good for keepers though, because when you're trying to find information online, it's so overwhelming to know what is up to date and what's accurate for you.
Well, accurate. That's the thing, and that's what this whole presentation is actually going to be about: is providing accurate information on these viruses that are out there now. So, and hopefully,
Dr. Google is not always our friend.
Yeah.
No.
Well, look, if you're getting new updates on pretty much the daily, then at least a once yearly kind of. I mean, there's going to be some things that are consistent, like quarantine. But I mean, different viruses, the quarantines are going to change, right?
Yeah, correct. Yeah, and that's one of the things I talk about in my presentation is. Our quarantine recommendations have changed considerably over the last sort of 2 years. We were talking 30 days, and then 2 months, and then 6 months, and now I pretty much say to people, don't even bother. There is actually no point quarantining.
Well, that's kind of what I think we were speaking about this last time. 900 plus days for PCR.
Yep, 950 days.
And that number's probably gone past there now.
It has gone past that. So I talk about a case that I had of diamond pythons that was over 4 years.
Wow.
So nobody's going to quarantine.
And it's not just pythons. You're looking at bearded dragons or common adenovirus. They can go their entire lives. I've got a positive one that's now 4.5 years old.
Yep.
But plenty of them live for quite longer than that, and
that's
an issue.
And is it symptomatic?
No, that's it.
You might get subtle symptoms that just come and go, but you never go looking for it if you didn't know it was there.
So serious, complete carriers, typhoid Mary style.
Exactly, exactly.
They'll have offspring, and then the entire clutch will die, but the adults will be fine, and because the clutch died, nobody will investigate, and then they'll breed again and sell them on, and then become another issue.
And just perpetuate the whole problem, which is the issue in America. That's what they've had over there. So.
Yeah, so they've got, I hear they've got huge issues in some of their captive populations over there, which I think we're going to be getting to eventually. But for now, I wanted to ask you about some of the reptiles down at Carrigal. I know up here it's pretty tropical. We've got a lot of pythons. That
you see a lot of pythons. So.
I'm pretty lucky. I get 4 or 5 a day brought in by you guys.
Exactly. I'm pretty regularly bringing pythons. We'll bring you the occasional brown snake and stuff. But look, I grew up down in the Dandenong Ranges in Ferntree Gully North. So. I remember if you're seeing the snake down there, it's unlikely to be non-venomous.
Well, it's going to be venomous.
Unless it's an escaped.
I actually have a friend of mine who's now a vet, reptile vet, works in Chicago, and he came and spent some time with me, and we went out herping, and I had to warn him, I had to remind him to say, if you see a snake, don't just pick it up.
Yeah, it's a little bit different.
It's a little bit different.
Like, can't you tell by the pupil?
Yeah. Or the lack of
don't pick it up.
pits.
And he's a very cluey guy. He's one of those really annoying people that knows a scientific name for everything on the planet.
I hate those people.
I hate him. I really, And the thing was, he knew all the scientific names for all the strange species that we were finding, and I'm going, that's a da-da-da-da, and he'd go, blah, 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 and I go, whatever. So,
it's a tiger snake.
It's a tiger snake, So we get plenty of those. So, so from a wildlife point of view, we get, by far the most common is the eastern blue-tongue lizards. So we would get 5 to 10 of those a week during the summer.
Largely dog attacks and things like that?
Dog attacks.
People just bring them in from their yards?
Yeah, they find them, on the road. It hasn't moved off my front step for 3 days, it must be sick. Yep. So they bring it in. We get some that come in that are quite a lot of ticks over them, so we clean those up and we get them back out. But mainly, dog attack is by far the most sort of common thing that we see with them. So
that cold turnover period down in Victoria is really interesting because you will get blue tongues that are coming right out of torpor, and they're like they look like they might be ill.
Yeah, and
it's just cold.
Yeah, they're just cold. Yeah, they just haven't been able to get under the log or something in time, that sort of stuff. So
yeah, exactly.
And a good majority of them will do the treatments on them and get them to a state where we can hand them off to wildlife carers and get them out. Sometimes it's a case of look at it, this animal's fine. Go and release it as quickly as possible, type sort of thing. So so blue tongues by far, and then the second most common would be eastern long-necked turtles.
Turtles.
So where my practice is, we're on a sort of just on the fringe of getting into semi-rural.
Crossing roads.
Getting clipped by cars. We've had a few rehab cases.
So we get normally I'd say we get about 40 to 60 a year come through sort of between November through to end of March about now.
Wow.
But this last period I think we only had about 10 or 15. So it was
I haven't had as many turtles this year.
We did not get as many. We did not get as many.
Did you bring me that turtle, the one that had been hit by the car? That
I did. I think I brought you one that ended up going into rehab here as well. Yeah,
that was that long neck that we got. She was in a bad state, but anyway, we're not getting many this year.
We didn't get that many this year. So
she did super well, by the way. That long neck, she put on a lot of weight. She got, she was really emaciated and horrible shell crack, but she did really well.
Yeah,
really?
They do. They're tough animals.
They are.
Super tough animals.
It took a while to get her feeding, but then once she did, she just
you did all the hard work.
Appetite just went Really awesome little creatures, those turtles.
Oh, they're tough.
Yeah,
that's why I like doing them. So it's very hard to kill a turtle. So it makes you look good.
If its heart beats once in an hour, you're right.
Yeah, you're right.
So reptile-wise, do you see like a lot of captive versus wildlife? What's
a good 95% of what I deal with is captive.
Okay.
So we
So you will be seeing pythons in that case.
We see heaps of pythons. Yeah. So we have a pretty busy practice. So we'll see 60 to 100 cases a week in reptiles. Wow. So it's pretty full on. And a small percentage of those are wildlife. We do a bit of work with some of the local snake catchers, and they'll bring in injured tiger snakes.
What they find,
So we get tigers, we get copperheads, lowland copperheads. Very rarely we'll get a little whip snake come through.
We get a lot. We get quite a few whip snakes up here with cat injuries. We've got the yellow-faced whip snakes that are super common up here.
I feel like you see them everywhere.
Cat
nice
injuries mostly for you, or?
Sorry?
You're getting a lot of whip snakes with cat injuries mostly, aren't you?
Yeah, and cats with whip snake injuries.
Goes
both ways.
Well, have you ever had that situation of treating a dog for a snake bite, and I'm treating so I've got my vets treating the dog that's been bitten by the snake, and I'm treating the snake that's been bitten by the dog on the opposite table, and you're trying to save both of them.
Angry?
No, it's funny. People, yes, they do. Mainly when you hit them with the bill. But I think they appreciate that where they live.
Trying to help both.
I've had specific cases where I remember them being actually quite upset by the fact that the snake was injured as well, and you do get that as well, which is nice. I mean, it's we're cohabitating, and it's just what happens. But we have to be careful.
It's good that people have that respect.
I think the attitudes are changing to some degree, and they keep building these housing estates where snakes were. It's part and parcel of living there. So. I need to accept that. So
look, even as a snake catcher, I over the last even 5 years, there is less people, with the shovel attitude. And I've spoken to other snake catchers who say that it used to be much worse back in the day, obviously. And obviously, slowly learning. Hopefully, we all are.
We're getting there.
Things like this help.
Now, I guess issue-wise for some of the animals coming in for captives, mostly husbandry-based issues a lot of the time for these animals.
pretty much everything's hydrogen based until proven otherwise.
Okay.
So we see a lot of respiratory infections and metabolic bone disease is still a common thing that we see. We see a lot of cancers, which aren't necessarily husbandry related. So I think it's now that we're
super underreported in some of the literature as well, as I understand. Getting better.
There was a new one paper just came out, the 2017 one that went and actually reviewed everything, and it's been. They say increasing numbers, but the same as with everything else, we're just diagnosing more. I think.
That's it. I don't think cancer is actually becoming more common.
It's just that we actually see it.
People are frequently diagnosed with better diagnostic tools.
There's more animals being kept. There's more people bringing their animals to the vets that actually can be treated.
Probably more people have access to vets that actually know what they're talking about.
And the animals are living longer.
Yeah, right.
You know, they're not dying of husbandry-related conditions early on in life. They're actually getting to an age. I think there may be some instances where there's a lot of genetics involved, where the original animals have come from a small gene pool, and you know you certainly see certain cancers in certain species. And I think has to have something to do genetically back to the base stock. I think a lot of it is that people are willing to come to vets now, and there are vets like Josh and I that see reptiles now. So
well, I mean, you guys went around doing, or at least as prevalently and as well known, even just a few years ago.
Well, it's taken me 15 years to get to where I am.
People are always looking for good reptile vets, and once they find one that they trust, they won't go anywhere else.
There's not many of us around, which is good for business, but it's bad for the general reptile community.
Well, hey, they know where they can find you now. It's a good start. So look, you've got housing and all these other issues. I guess, do you see a lot of injuries as well, or?
Yep.
I mean, I've heard of some pretty silly stuff.
Got a great photo on my phone right now. There's somebody. I had the two children's pythons brought in to me at the same time the other day, and in one bag, and he opened the bag, and one was inside the other one. And he said, take a photo because you need to share that with people so they know not to do that.
Not to bag.
And he goes,
what were they coming in for?
Just health check.
Oh, great.
And they were both fine in the end, by the way. They were absolutely fine, but they can end pretty badly. So they do things that they shouldn't,
and it's it's silly mistakes like that. Probably the biggest traumatic ones for me is bearded dragons being habitated, so keeping them in together, and you lose tails and legs,
fingers,
all that sort of stuff. So
I've got two dragons just over there, and male and female. Still won't house them together. Just for the fact that they will nip each other's toes off.
Male, female, does not matter. It takes literally a second, and you have a crushed skull.
And they're perfectly happy separate as well.
And the reality is, they look at it in the wild. They don't live together in the wild.
No, they don't.
We stick them in a 4-foot enclosure and hope they live together.
I'm actually treating some turtles at the moment. It's similar. It's unfortunate. I mean, big pond, but males and females together. And the fact is, the males are after the females, and that's more time of the year than they normally have it, and that's putting stress on them. And it's the stress that causes not just injuries but disease.
It sounds stressful.
Well, having lots of men chasing them.
And look, all of these stressful things can lead to disease as well.
That's right.
So look, we should start speaking about reptile viruses here in globally and in Australia. I guess there's not so much known amongst some of the general community, reptile community, about some of these viruses and how prevalent they are, how bad they can be, how bad they can spread. But you do occasionally hear stories of like whole collections completely lost and wiped out, both here and abroad. Are there many like global risks for reptile keepers that we have to keep an eye out for? Everywhere?
Yeah, I think we do. It's I mean, there's viruses that are here that because of the way they keep overseas, it would be spreading a lot faster than over here as well. I think you might have spoken to people sooner and know even some more stuff going on. But that's always a big concern of mine. It's you go to the shows over there, like we're going to have the show here where you don't have contact with animals, but I've been to shows. We went to that one at the conference.
They're like, just importing and exporting viruses.
Last year
I'll just take that one out, put it on my neck, go to the next guy's thing, and then go to the next one. And that's a recipe. And if you've got 3 years of lag time before diagnosing that, then they're going to be breeding more, and it's going to go very.
See, my attitude at any expo was like hands off.
Yep, correct. Which is
so hard.
Opposite. Really hard.
It is very difficult. You got to keep your hands behind your back, shove them in your pockets.
In Dallas last year, and just so happened that the Reptile Expo was on the week before our conference that we're at, and it was really interesting to see the different attitude at that show compared to the expos here in Australia, and I think we've got it right.
Yeah. Minimal contact.
Everything here is very clinical.
If it's,
I like
it. If they're display animals, they're on display. You don't touch them.
Which
you only touch demo animals.
Correct. Yeah.
Which being okay.
It has its negatives.
Yeah, exactly.
It has its negatives, and people will complain. I can't handle the animal, so I don't know what its temperament's like, or I can't check it over and make sure there's a problem. So
if you get to Brisbane, you can't even.
Yeah,
we can't sell it anyway. So
but
that's really not the point. It's really to display.
Yeah, but it's a very different attitude, and I think we have it right here. We're trying to minimise that. In saying that, you're still in a room that has
however many animals. Yeah, which might have respiratory.
Correct.
And we don't really know how some of these are spread either.
Spread. So from an expo point of view, that is a concern, and then certainly from a private keeper point of view, taking something back and putting it into a collection is that has changed. Well, hopefully it's changing with the more knowledge we know about these things.
And what about, I guess, things like either permitted or even illegal wild collection? I mean, Australian endemics are so popular that, with big native wild populations, you have viral reservoirs.
Correct.
Viruses that we probably haven't even seen in captive collections before.
Yep.
So, is there a chance that we're just going to keep seeing new things pop up?
At the start, I mentioned about how I got them brand new. Literally, the conversation I had with somebody yesterday was exactly that. There is a virus living in a resident population in the wild, and we were discussing. Her and I were discussing the possibility of that virus actually being brought into captivity, and in the wild population, it probably doesn't do a lot because these animals are evolved with it. They cope with it,
and they probably have other viral loads.
They do,
and so those viruses are running their own red queen
effect. The stresses that those animals are put on are very different. You take those animals. Take the same species, you put them in a captive situation with that virus.
Yeah, they can't get away from their fecal matter, for example.
And so you get super infections, you get all sorts of problems, and so that's a real thing, and that's a thing called Bornavirus is what we're talking about, which is one Bornavirus, which is one I'd not even heard of. Townsville, that I'd not heard of until recently.
Wow.
Yeah, you flicked me the paper because it's that new, and that's the only way we find out as well. It's one person finds out, we tell the next person, and then finally we can disseminate it.
Yeah.
But you look at arenavirus in boas versus pythons. The difference in symptoms, and some evolve with it, so they're much less likely. Boas tolerate it very well. Pythons die.
There's a real species differentiation across some of these viruses,
and they're not far apart.
No.
So it's a big.
But you mentioned about whole collections going down, and that has happened. I've dealt with that. Where you go in and test. A collection of 20, 30 animals, and a very large percentage of them have one of these viruses, and you have to say to a client,
sorry.
In fact, and again in my presentation, I've got a slide of a group of snakes in pillowcases, and it's my single biggest, and I put in liked in inverted commas because it wasn't a liked thing on my Facebook page, the biggest thing I've ever put up there, because in this, it's simply a photo of about 10 pillowcases, and I discussed that I'm just about to euthanize all these snakes that are in this picture because they've all got sunshine virus.
Wow.
And this is what happens, and if you don't use quarantine or practice good quarantine, if you don't appreciate that these problems can exist.
Very severe consequences.
Yeah, I was going to ask, with the whole collections, is that mostly through equipment sharing, or the person handling it, or
so
everything?
It's a bit of both. So in that particular collection, there was a large number of people going in and out of that facility, and with no disregard, not disregard, that's not the right word. They were washing their hands and that sort of stuff, but it only takes one little break, and I've heard of, and I have no idea if it's true or not, but I've heard of rumours of whole collections going down, and particularly one that was a green tree python collection, and the guy had a whole setup of quarantine, that sort of stuff, and he made one mistake, transferred one animal,
That's
and
a big loss.
Lost everything, and so the numbers that are thrown around are $150,000 worth of animals.
I wouldn't be surprised.
Bornavirus too.
Bornavirus. And then if you look at some of these viruses, if you actually look at the published data, so there's a virus called Virus, and that wiped out an entire Swiss reptile facility, 100 animals, killed them all. So it happens. It certainly happens, and
share breeding is the one that scares me a little bit. It's the giving of animals,
breeding loans
to breed or share, and then
without
now you've got two collections, not just your own.
Yeah.
And it's your lack of understanding that that's not. And
you've got a pretty snake, and I think my snake would do well with your pretty snake. So I'll give it to you for a week.
Rather than putting them through health checks or quarantine.
Well, the problem is that I give it to you, and then I'm your mate, so therefore you mustn't have viruses because we're best mates.
Oh, because we trust each other.
And then
it doesn't look sick.
Bang. Yeah, it doesn't look sick.
And it doesn't look sick. And
snake comes back after the breeding loan, you put it back into the collection with no quarantine, no testing, no whatever.
Yeah.
And all hell breaks loose.
Do you think also with like the amount that I feel like in like the reptile community people sell and buy and everything?
That's actually and that's a whole separate issue for me, and I know Josh, we've discussed this as well as we do. The exchange and transport and movement of animals around this country with absolute disregard for their health
and safety,
their identification. I'll buy. You could be in Melbourne and buy an animal from someone up here, and then you get the animal delivered and you open the box and it's completely not what you ordered, and then all hell breaks loose with that.
Frequently.
I've had people that they like ordered like a male bearded drag or something, it turns up with a female like full of eggs. Yeah. And they're like, oh, I'm pretty sure you sent me female. I'm like, no, it's male. It's like, well, there's eggs in this box. So.
I've heard plenty of cases of venomous snakes being misidentified.
Wow.
And that's a whole other ballgame, isn't it? So I have I think the problem with us in Australia is that our hobby has actually gone and expanded faster than
the
legislations and the policies, and you would know doing what you do for a living, than our policies have actually been able to keep up with.
Right.
And it's scary.
It is.
It's really scary. So.
Well, look, especially with the amount of disease risk.
Well, you can track these viruses from where they originally were found, and you know that they're going to spread across to other places.
There's nothing worse than having to say how it could have been prevented.
Yeah.
It could have just, because I'm always asked, what could I have done? So, well, that's the breakdown point, and it's so simple usually. And that's a problem.
Yeah. Well, that must be a struggle for you guys being able to see some of these problems on the horizon.
I
suppose that's the point of the talks.
And we were talking in the car on the way from the airport here, and the thing about my talks is it's great. We've got lots of people coming. What's actually scary for me is the people that aren't coming, and the people that are coming and at least know and have heard about virus infections and those sort of things.
And that's why they're interested.
That's why they're interested. It's the people that have never heard of it, and how do we get to those keepers to say, come on guys, you need to be aware of this, because whether you've got one snake, one lizard, or a large breeding collection or whatever, you need to have at least a base education on this sort of stuff.
And it doesn't necessarily just affect you; it affects other keepers. It affects the
and snowballing from that is let's look at a worst case scenario. We get a viral infection that goes through collections, that sort of stuff. There's nothing to stop government departments turning around and saying, actually, you're putting at risk wild populations. You're now not allowed to keep reptiles.
Absolutely.
Now that's absolute worst case scenario, and it should hopefully never get to that point. But there is always that risk that can be take our it's not a right, it's our wants.
We are kind of out of privilege.
That's exactly what our privilege of keeping reptiles is at the whim.
We have to do it well.
Government. Who can take that away?
And that does depend on things like not actively further spreading sunshine virus.
Correct.
And doing our best to minimise this.
Because I would hate to think of the day where sunshine virus potentially gets into the wild population of carpet pythons around Brisbane. That's a scary thought. That's very scary.
All right, well, look, let's move on to snake viruses themselves and sunshine virus.
Yep.
So let's start there. It's a python paramyxovirus.
Correct.
Is it a Paramyxoviridae? Is that a family, or?
So it's actually now put in its own family. I can never get this taxonomy right. I think it's its own family, Sunviridae. So when it was first found, it was put into the Paramyxovirus Viridae, but it's now actually been further classified into its own
genus within Paramyxoviridae.
Yeah.
Okay.
Is that a contact spread?
So it's spread by contact, fecal,
we think
fluid.
so.
Well, we test positive orally and through cloacally.
It's found in body sprays. And I actually had done some work. Actually, that collection I mentioned about, the ones where I was about euthanasia, I actually went back to that collection and we tested hides and the floor and the doorknobs and the litter and that sort of stuff, and we found viral DNA in the environment. We
still don't know if it's active.
Correct. That viral DNA may have been dead, and it might not have been able to infect other animals, but what it told us is that these snakes are shedding the virus into the environment. So.
And can it be aerosoled as well? Can they breathe it out and respirate?
Most likely what it is.
So like if you're cleaning
airborne,
say water or something, and there's the feces in there, and it's actually aerosolizing that. But in water droplets, I'm not sure if we know if it's aerosol, if it's airborne or if it's just aerosolized
in those liquid.
In water droplets is what I think.
Don't quote me on this, but I think that is something that's being looked at the moment. There's some work being done on that. So.
All right, and it initially presents with issues balancing and movement with like kinks in the neck and
not
body
necessarily.
posture. So is that some of the time?
It's one of the symptoms.
Yeah. One of the symptoms.
Yeah, it's quite varied. So I tend to find sunshine virus is a neurological disease. Yeah. A neurological virus, and so these snakes, there's this classic term called stargazing, where they'll sort of be cruising along and then they'll just arch their head back and literally look at the ceiling, and they can't control their movement. So it's a real something goes wrong between the brain and the body.
Right. Now there's some snakes that will lift their head up as they come out of hives.
Yeah, that's fine.
But it's very arched.
Correct.
Back.
They go right over and they stay like that, and they'll lose their righting reflex. So you put them on their back, and the snake should immediately turn and flip back over. It's not a natural position, but they'll lay there. But the symptoms, I've seen them with just mild head tilts, and
I've seen them with just mild respiratory signs.
Yeah,
and I've seen them that's the problem
with the mouth gaping.
A little bit, and they'll just have a few bubbles. And both of us have seen them with absolutely no signs at all.
Right. So
I think it's the respiratory. And I think the biggest problem I have is that you've got sunshine-borne coronavirus or nidovirus, so we're reclassifying that, and that they all have respiratory signs.
Yeah,
and and Borna and Sunshine have neurological signs.
So they can have neuro-respiratory.
Correct. Yeah, and you can have a snake that has a bacterial infection, pneumonia,
that can present the same way,
exactly the same way, but doesn't have a virus, and so you can't look at a snake and say it's got
this.
So RI is probably my least favorite term because RI is a symptom, and it's a symptom caused by something which we have no idea when we look at them, and so sometimes you can treat them and they'll get better. Usually sunshine virus will not get better. There have been cases reported where they were clinical at one point in their life, and then like a year and a half later they became...
Yeah, they're very anecdotal and maybe
very
not the best sources.
I've
never seen one get better.
No, I've never seen one either.
But you mentioned last time as well, last time on the show, 900 plus days that they can present symptoms and be...
So asymptomatic for 900 plus days, and then all of a sudden show signs. But that's because they were tested the 3 years before.
So I was involved in a study that there was 4 vets across the country testing 5 snakes from 5 different collections.
Mm-hmm. Right.
And the owners of these snakes were, and I said this the other night, is we can't publicly recognise these people because there's a stigma with having viruses in their collections. But these people, what they did for us as researching...
It's actually quite incredible
they put the rest of their collections at risk of doing this because they kept these snakes that were positive in their collections admittedly separate and that sort of stuff. We were testing these snakes at least every month, and they were testing positive every month. And what happened with 4 of those snakes was they got to about 7 to 12 months after they'd been tested, and almost overnight suddenly developed neurological syndrome.
Wow.
And you sort of, I question and go, what is it that changed it? So they did that. And secondly, these animals were completely symptomless until that thing. One of these snakes was euthanized at 950 days from the time it was diagnosed. And it was probably positive for a period before that. And when it was euthanized at 950 days, that snake was still perfectly healthy. The only reason it was euthanized is the guy had said, the owner had said, I've done enough, time to get this out of my collection. So how long that then kept
going. So he did that to not risk the rest of the collection any further.
So
he's done with
after 3.5 years or whatever, whatever 950 days and years is.
You can't ask too much more.
You can't ask too much more than that. So, but that snake was not showing any symptoms, yet it was shedding the virus and was potentially a source of infection for the rest of animals. Right. And that's why quarantine now is completely a waste of time as far as I'm concerned when you're dealing with viruses, because you can have an animal go that long with no symptoms. How are you going to know?
Yeah, it'd be nice to know what the actually pre-patent period is for shedding.
You're correct.
Because we don't really know, and we give these recommendations of test them when you get them and then test them 3 months later. But there may be the odd one that won't test positive for 6 months, and they're the ones that I think will slip through.
So from the time they're infected to the time they actually can be tested, what is that time, and we don't know that. You know, we don't know that. So
still a lot to find out.
There are people coming to the talk tonight that are actually willing to say they've got it. And to talk about it. And they've contacted me saying, you know, we hope other people do too, but we're going to. Because I think it's very important to get out there so that we understand why it's happening.
If you shake their hand, bring some disinfectant.
we'll have lots of F10 disinfectant, that's the stuff. So,
now, look, I mean, this is a fairly new disease as well, so it is very important to have these people coming up and, you know, the more data, the better, and all of this. I mean, 2012 was it?
But it was 2008.
Early in that, so it was 2008 was the third. Yeah, and sunshine virus is probably the virus that started all this in Australia as far as people going, oh, whoa, hang on, what's going on?
2012 is when I became aware of it from the Tim Heinemann paper.
Yeah. So the original outbreak case was 2008, but I also know that there have been snakes samples tested from as far back as 2006 that tested positive. Okay. So the virus was around for at least 2 years before the original population of snakes were diagnosed with it. So, and then it's gone on from there. But it was probably the first virus everybody went, you know, when it came out, what it was, and then since then we've had other viruses identified.
Well, there was one of the original papers when they said they thought it was inclusion body disease, but it ended up, really hindsight, fairly likely that it's this other paramyxovirus virus.
So there's been other viruses that have been touted as being in Australia, but
it usually ends up being sunshine or some other paramyxovirus.
It's either ended up that, or you know, the reality is technology improves, and back when some of these viruses or some of these cases were presented or published, the technology wasn't there to actually identify the virus. There are other methods of identifying that. There was probably a virus involved, but
it was probably done by cell culture.
If we could go back, correct. If we could go back now and do DNA, PCR testing, that sort of stuff.
It was always done by histopathology.
Yeah, they'd always probably find something different. And then since sunshine virus has come, we've now got at least 2 other viruses in snakes.
So obviously the genetic testing, the reverse transcription, PCR,
all that. PCR has revolutionised the way that the researchers.
Real-time PCR has really revolutionised.
Absolutely. So sunshine virus causes. Well, it's a neurorespiratory disease, as we mentioned, and I hear it leads to hindbrain white matter spongiosis and gliosis, extending into the grey matter, and with actual full-on necrosis of neural tissue in some cases. So, like, literally rotting the brain. Is that just when it gets really severe, or?
Well, it goes from not severe to really severe very quickly.
And when you see these samples of these brains on the histopathology slides that the labs are looking at, it looks like somebody has literally got a shotgun and punched holes in the brain. It just eats the brain away.
So it's like
I said it kind of looks like a mad cow disease. Is what it looks like.
I was going to say Al Capone and his syphilitic brain all being eaten away by
Swiss cheese. It's just got holes in the tissue all the way through.
That's what distemper does too.
A lot of viruses will do that, but sunshine virus is particularly nasty that way.
Right, and of course, you know, being that's a respiratory thing, you get that bronchiointerstitial pneumonia. Any new developments in sunshine virus that we need to be particularly aware of as snake people? Other than the fact that it's 900 plus days asymptomatic
that's
carrier.
Probably the big one. It's still out there. I literally had a case that I diagnosed yesterday.
Wow.
So you'll hear people sort of saying, it's
gone.
It's gone. It's
when is the virus ever really gone?
Well, that's right. The vets are just making this up. It's not as bad as it is, and it's not as common as it was, and that sort of stuff. I'm good, thank you. But no, we still get cases. I'm still diagnosing 1 or 2 a month.
That's quite common. Well, I mean, you do get a lot of reptiles.
Yeah, we get a lot of reptiles, but it's not. I wouldn't say it's real. It's not as common as it used to be, and I think that's because people maybe are learning, but it's still around. So.
All right, and so some new viruses out there. Reptile Virus. Not exactly new, but. The cause of, we believe, Boyd inclusion body disease. You get these within the cell intracytoplasmic inclusion bodies, basically masses inside the cells, and this is spread by snake mites. Is it? Or thought to be spread by snake mites? Something kind of like Lyme disease for snakes.
It's never been proven.
Never been proven. It's just a suspected vector.
And people like Josh and I are always on the lookout for collections that we diagnose viruses in that have got mites. Because then we can collect the mites and get the mites tested. But I've never had one yet, so
Well, I understand that it's been at least.
Although nobody wants to admit they have mites.
Yeah. But didn't we only start seeing mites like 20, 30 years ago, or is that something different?
I'm not sure. Honestly.
Yeah, I don't know when the first.
No.
To be honest, yeah.
First case of snake mites. That's interesting.
In Australia, I mean.
Yeah.
Like overseas a lot.
It's an exotic. The mite is exotic to Australia. So.
Yeah, I thought.
Is it from the US as well?
Oh, probably. Everything good comes from the US. Doesn't it? So,
I can agree with that.
Yeah.
There we go. Yeah.
But yeah, probably. But I actually, I don't know.
I remember someone telling me
that's going to bug me all day, and I'm going to have to go and look
at
it
tonight. Josh gets that. He'll go and look it up now, and I'll just go, that's interesting.
I'm probably right, so don't worry. Don't look it up.
All right. Okay. No, well, if anybody else out there is listening, feel free to check us on any of this stuff, by the way. Feel free to write in.
Yeah.
So how much virus do we have here in Australia?
Zero.
None at the moment.
Oh, good. I'm glad we both said zero. I certainly
I waited till we hand up.
Yeah. So it is a paramyxovirus, though. Simply.
It's a paramyxovirus. So that was the original virus that the researchers were looking for when they discovered sunshine virus.
That's right.
And that's how the whole story
I have
happened.
tested for it still because I've had these weird cases that don't look anything like any other sunshine, which is how I found my first Bornavirus test.
Yep. Right.
I was looking for either Furla or actually I was looking for Furla, Arena, or Sunshine.
Just going broad.
And, well, no, I did Sunshine first, came back negative. Then I did Arena, came back negative. I said, I'll try Furla, came back negative. Now I have no idea. Oh, and that's when I got Borna because it was suggested to me by the testers when I found Borna.
So
right.
And that is what's happening.
Yeah, right.
We're accidentally finding viruses looking because we get presented with cases that you look at them and you go, it's got to be a virus here, and it tests negative for everything, and you go,
it's always a virus.
And we're very lucky in this country. We have a researcher that will go hunting.
He does,
and he finds these things, which
that's fantastic.
Is a problem,
amazing.
But,
the problem is now, what do you recommend? And I've asked him specifically, said based on what he sees, what should I go for first? And if it's negative, he says go for this, and then if it's negative, go for this based on the symptoms you're seeing. And sometimes we find something new. That's how I found Microsporidium. That's how I found Corona in these collections, Borna. It's and now we're getting a bit better at knowing what to look for, but it's changing.
So we do have Bornavirus, which is has been found in lapids, mammals, birds.
Yeah.
There's Nidovirus here.
Pythons.
We have Nidovirus
in our lizards as well.
Yeah.
So are these just...
actually that night we're talking for about wild that Nidovirus, shingleback Nidovirus 1 as they call it. And
the newer one?
What worries me is they've named it 1, which means that they think there's probably others. So,
there's going to be multiple.
There's going to be multiple. But that's an example of a virus that's come from the wild into captive
populations.
It's a wild population.
From the population that Mike Ball was studying with the...
Yeah, in Perth, around that area. And that's come into because they have the permits to be able to catch and bring them into captivity.
Right.
That's how that's probably got into captivity. And then it spread across to the East Coast because of the transport of animals across. And so you've seen it up here. I'm seeing it certainly down in Melbourne.
And pretty similar to sunshine virus in the neurorespiratory.
It's a respiratory disease.
So they basically
So the old name for it was bobtail flu.
Okay, right. And huge amounts of mucus and stuff.
Yeah, they get bubbles from the eyes. They get the sniffles.
But then you get the thing we have Nidovirus in pythons.
Yep.
And we've got Nidopythons in the US, and that's wiping out collections, and here it's just respiratory disease.
Right.
And they actually are recovering from it.
Yeah.
I don't think I've had any die from it.
So the Nidovirus in snakes is a really interesting one because it's the hot topic in the US at the moment because it's killing snakes.
Right.
Technically it's coronavirus.
It's a respiratory disease, and yet my understanding is that they're all Nidoviruses. They're all in the same family, but there's slight differences in them.
Yeah, small.
So we don't have what they have out there, but it's killing green tree pythons. It's killing them.
And you've got to wonder if there's been evolution, so they're more tolerant here versus over there.
Yeah, of course. Or maybe they have, they bottleneck the population at some point. And they have low genetic diversity, low amount of major histocompatibility genes. So,
but it is amazing the difference in it is wiping collections out over there, and over here I've never had one. Well, it takes a little bit more, but it seems to resolve.
I've got a black-headed python.
But then I checked the next year, and they're still carrying or shedding, but not showing signs. And it seems to when you cool them down. So stress with all of our viruses, I think stress seems to be the biggest one. You go from normal, something stressful happens, whether it be breeding or cooling or moving, and then all of a sudden they within a couple of weeks sometimes they're getting sick.
What about in the shinglebacks? Is it usually fatal?
No. Well,
chronic.
They get chronic and they get a wasting sort of syndrome. They lose a lot of weight in their tails.
Can't smell, you don't eat.
Yeah,
right.
And they
that's what. So
can
you it's like a longer term.
Sorry? Correct.
So is it they're getting a lot of mucus and they can't smell, they can't feed? Can you assist feed them in?
And that's the treatment for it. It's basically supportive therapy. You're syringe feeding them. You're giving them subcutaneous fluids. You've got them on antibiotics. And a good percentage of, I don't think I've had a shingleback or a blue-tongue lizard die from nonviral. That they get over it, but the next year, and that's really what we're finding now. We know this virus exists. Is these snakes and lizards that I'm seeing that every year they seem to come in with a recurrent respiratory infection. We're now going, hang on, let's maybe test for this virus. And what do you know? Look at that.
It just keeps on popping up.
Yeah, and
what I'm interested in is when they clear it, how many of them are clearing it or they're not shedding, and then next year are they shedding again? Because it makes it easier to make a recommendation as, okay, it is gone. We get rid of colds, and then so I that's one of the things I'm looking at a little bit.
And
that's a really clear
cleared out. That's a really new virus. Like we're talking less than 12 months. And so we know less about that virus
than we do
about
all
the others. And so
all right. Well, what about in lizards, adenovirus?
Yep.
So it's a broadly infectious group of respiratory virus, which includes humans. Humans get adenoviruses.
Yep. Well, that's how it got its name. Adenoviruses is because it came from adenoid tissue. So
adenoid tissue in humans. Oh, right.
Yeah. So it's like
adenovirus, right?
Love it. Or that's in America they call it adenovirus.
And very common over there in US lizard collections, from what I hear. But is it found here?
I think they said something like 80% of breeding collections are carriers.
80 something percent of the breeding population.
That's what I've heard.
Bearded dragons over there. What
it's
about
very. I probably 4 in the last 2 weeks.
Wow.
Yeah.
Okay, so is that more prevalent than it has been before?
It's just not being tested for, in my opinion.
So it's again a detection issue more than.
I think for a long time we didn't know it was here, and then it started getting reported, and we've all started going.
There's also a number of adenoviruses in lizards, but there's a specific agomid adenovirus. So it's the one that's really just the agomid lizards that seem to get it.
So get it very specific.
So agamids, the dragon lizards here. Water dragons, bearded dragons.
But we're testing for it more, so therefore we're finding it more. If you go looking for stuff, you find stuff.
And what you find is that, I think I was saying before, a breeding pair, you'll have your clutch. They'll all die, or some will die. And if they're not tested, we don't know what happened. And then those 3, they actually will become carriers in many cases. And then they will perpetuate.
And it's a highly infectious virus, and it's a virus that lives, unlike something like sunshine virus, which probably doesn't live very long outside the hosting environment. Adenovirus is something that can live 18 months.
Right.
And
so if you haven't bleached the thing and you
put
something else.
Scenario is you've got a female who's positive, lays eggs. Those eggs on the surface are covered in the virus. That virus can live
in the substrate.
In the substrate in an incubator. The babies hatch. What's the first thing they come into contact with? Surface of the egg.
Nightmare.
Infected.
Well, and they've got 6 other clutches in there or whatever.
And how switched on is the immune system on something fresh?
So then you go into things like you're not only doing. You might have quarantine set up for your bearded dragon facility, but everything funnels into an incubator. And you got one incubator.
That's not quarantine.
That's not quarantine.
Yeah, no. There's always.
I just wrote a thing on quarantine because it's if you actually look at quarantine, it's actually impossible. Quarantine is impossible because it does need to be in a separate facility. And you basically need to. Nobody else who has handled a reptile, usually in that day, should ever go in there. Right.
I would say you can have a degree of quarantine control, but
you can have a degree of risk reduction.
Okay, that's a much better way to phrase it.
And I'd call it, I'd say it's more like isolation as opposed to quarantine.
Right.
So you isolate something to keep the disease out. You quarantine something for long enough for the disease to show itself.
Right.
And if it never shows itself, how do you know when to stop quarantine? And that's what it comes down to.
And I've been to a full quarantine facility, a vaccine manufacturing plant. And you have to go to that level, and nobody is going to do that level. You physically can't. You can't.
No, nobody's going to work in a PC4 container lab in their home.
And that's quarantine. What we're talking about is isolation and risk reduction.
Absolutely.
And people need to understand that. So.
But there is definitely steps that people can still
absolutely take. There is absolutely steps about reducing risk.
Yeah.
You know, and you can reduce it very, very far.
Yeah, yeah.
But to refer to you know, just keeping animals in one room as quarantine when there's no
depends on their habits.
Yeah.
Not the room itself.
Yeah.
So if they it has really little to do with the room so much as to be what their habits are.
Yeah.
Right.
And a room in the same house is
well, if the airflow is connected.
Yeah.
Yeah.
I couldn't say be quarantine.
Yeah.
We do use the term still, and we use it loosely.
Yeah.
But to kind of put a seriousness to it, I think.
Right.
Yeah, I agree. But it's probably not the right. Yeah, I like your word of isolation.
Yeah.
Well,
an isolation facility.
I was writing this thing, and I went back to the definitions, and I went to this CDC, and I said, all right, what is quarantine?
Yeah.
And I said that is quarantine versus isolation. You can be isolated in quarantine, but isolation doesn't necessarily mean quarantine.
Yes.
So I think that's what I had to.
Yeah, that's a good point.
All right. Oh, look, moving on. Just I guess briefly before we get into a bit more on quarantine itself. Turtles and crocodiles.
Ooh.
Yep.
There's a Nile crocodile pox virus.
Yep.
Is there many Australian crocodile viruses that we need to worry about?
So they've got a herpes virus.
Yep. Okay.
And the problem with that is, well, not the problem with that, is it's probably one of these viruses that has been in the wild for many, many years, and so most of the crocs that are probably coming into captivity out of the croc farms have got herpes virus.
Right.
And don't show any sort of symptoms.
Carriers.
As carriers, yeah, absolutely. So very similar to herpes virus causes cold sores in people. You get run down and stressed, you get a cold sore. Same probably happens in the crocs. And so the difficulty with diagnosing that one is it needs a swab from the back of the mouth.
Have you ever seen one with corneal ulcers like herpes virus? You have. I've seen a few corneal ulcers which I thought were water related, but
in crocodiles.
In crocs, they get a lot of eye and skin problems.
Okay,
yeah,
that makes sense.
I've dealt with a
mucus membrane that's constantly exposed.
I got called in because these crocs were getting eye problems.
Was it water? Or did you test for herpes?
Ultimately, on necrops in histopathology samples, we found that they all had herpes virus. So,
keep that one in mind.
Yeah, so but that in Australia we're lucky. There's another one called West Nile virus and a few other things, but we're pretty good here in Australia.
Right.
Well, what do you do when you find out whether they have herpes virus?
Well, to be honest, if the animal's well otherwise or mildly affected, is you treat them supportively and their immune system will basically cope with it. You fix the husbandry issues that ultimately resulted in the animal becoming stressed, and that will fix it. So,
do you remove warts and lesions the way they do in people? No.
No, you can't, because the herpes virus lives in neuron engine.
Yeah, it lives in nerve cells.
So, and what happens when you get stressed, well, people who get cold sores, they feel a tingle, and that's actually the activation, really, I believe, at that time.
Of the nerve?
Of the nerve.
It's the virus replicating in the nerve.
Yeah.
All right.
And it's, that's part of the issue.
But there are other viruses, things like papillomavirus, like green sea turtles, for example.
Well, they also get the herpes virus.
It's thought to be a herpes virus that causes this papillomatus thing, and they come in with lumps all over them, and they're cutting those off. So
I was in Hawaii recently.
A bit easier to swab the back of the green
well, not necessarily. No,
they got that big.
Well, you just biopsy them off, and they take those and they grind them up and make a vaccine and re-inject it in, and it kills the. It does work.
But Josh was.
I was in.
That's how the first vaccinations were done with the smallpox, right? They took the people who are surviving the smallpox. They actually took the scabs and ground them up.
Hey, they're doing it for cancer now. Taking the cancer, grind
it up. Very gross, but very successful.
Re-inject with the cancer cells as a certain antigen, and the body attacks the cancer. So they're working on those things now too. So autologous vaccines are big. But anyway,
that's phenomenal.
I was on the beach in Hawaii. And
it doesn't work very much.
No, never. And no, don't ever work. And we were walking down the beach, and there was a turtle there, and we thought there was something coming out of the mouth, and obviously not allowed to approach them, but I did take it upon myself to go have a quick look, and there were papillomas all along the mouth line, and what will happen is they'll grow over and they can't eat, and so they starve to death. And it's very common in Hawaii and Florida, Texas in the Gulf there.
So like swelling of the mouth, stomatitis, and all these secondary conditions leading to...
It's actually the growths. They grow over, and over the nose, over the mouth, over the eyes, and then they starve.
So they are actually removed. They use a laser surgical.
And you can, and they grow back.
Yeah, they'll grow back.
They'll grow, but you'll get a bit of relief and enough time for them to eat.
Yeah.
And there are antivirals, acyclovir, famciclovir, and things like that, but they're not been tested very much in reptiles.
And it's a bit hard to do them in green sea turtles.
Oh, yes.
Wild population. But there's a lot of talk about like global warming, whether that's resulting in the
increase
virus
in
being able to replicate faster, and the pollution and the stress of plastics and all that sort of stuff.
And if you're putting if you're decreasing the water quality, then obviously the animals are going to be more stressed and less healthy, and then those viruses are going to be more of an impact.
Yeah, so I think it's multifactorial of what
as it is
results in these things. So absolutely.
All right. Well, look. For anybody who's listening at home and is getting a bit confused, don't worry. There's many other reptile virus groups out there. There's iridioviruses, rhinoviruses, papillomaviruses, rheoviruses, retroviruses, and more. So reptiles being a pretty broad taxonomic group.
I think they've been around for so long that these viruses have really adapted to them.
Yeah. So how do you guys stand? Is it just the networking that you guys do?
We just make it up as we go along.
All right, well, invite you guys back.
Lots of reading.
Yeah, I can imagine.
Conferences, and knowing people who know what's
knowing
happening.
People who know stuff about viruses. So,
and then constantly going into
and that's the thing is, Josh and I are not researchers. We are clinical vets, and there is a whole bank of people out there, and many of them are vets that are doing research into these things. And these are the guys that they come to the conferences and they present their work, and we're sitting in the audience going, "Oh, a new virus," and that's how we keep up on that. And thankfully, over the years, we've got to know those people personally, and
we get the heads up.
Yeah, we get the heads up on it.
And you look at how we can contribute to it. I mean, the numbers of animals we see, wild and captive, and
you can definitely provide a lot of data.
Well, we can provide the samples.
Yes, that's right.
The reality is, both Josh and I have sent lots and lots of samples into researchers. That has resulted in expansion of the knowledge on that, and I'm pretty proud of the fact that I've had a lot of involvement in doing that and helping to get more knowledge on them.
Yeah. So, well, look, it seems like we are slowly starting to learn more and more with new research into viruses, but it's still a lot to deal with, and that's not even taking into account all the other stuff you got to deal with, like bacterial and fungal infections.
Yeah, the fungal ones.
Yeah.
More and more fungal infections as well.
Yeah.
Watch this or at least being we're becoming aware of more and more now with new diagnostic methods and things like that.
Yeah, there is a lot of stuff happening.
Yeah, Josh sees a lot more of that up here. And
I think it's a bit warmer and more humid up here.
Possibly.
I think environment has a lot to do with it.
Yeah. Even some of the bacteria that do the the fungal type appearing bacteria.
So just being warmer and
dermatophilus is probably the big one.
It's
one that's high humidity.
Which
dermatophilus congolensis. It looks like basically they're like little abscesses on the skin, basically, and it's a bacteria that has very filamentous, and it seems to. I actually thought initially it was going to be fungal, and it wasn't. I mean, it's a well-known one, but in this area we do see a bit of it.
And whereas it's fairly rare, saying that, I had 2 cases last month, but it's still pretty rare, and I think it is the environment. It's the warmth, it's the humidity.
Well, we are in the subtropics. It's fairly likely that we're going to be seeing
and
fungal.
And CANV, of course.
Yellow fungus.
Yeah, we were speaking about yellow fungus last time. Has there been
as the Opsis ricei?
I've yet to see a case.
You're yet to see a case, but we're seeing
4 now confirmed, and I think they were the first ones in bearded dragons because it's been seen in eastern water dragon. Eastern bearded dragon, Pogona barbata.
I think I was going to say,
but not Viticeps. And these are in private collections, and 2 from one collection, and now 2 from other separate collections. One I'm presenting at the thing tomorrow. I've got a poster of it.
Oh, cool.
And it was actually an intranasal lesion, which we've cleared and used antifungals internally, and it's actually seemed to get rid of it, because it's very difficult to get rid of.
It's invariably considered to be fatal.
Success 1.
Yeah.
We've got one.
Small steps.
Yeah. Well done, dude. Well done.
He saved something.
Sometimes it feels like that.
It is a hard slog. I know, but dude,
by the time
honestly,
they come to you and they're sick, a lot of them are dead. And that's the problem.
Yeah. Well, look. That being said, a lot of the time, even providing euthanasia is. It's still a very important service.
It's a good service.
Yeah. It's very, very important.
Yes.
How about you, Shane? Do you deal with a lot of non-viral stuff down there?
So as we were talking about, we see a lot less of the fungal things. We still see plenty of bacterial infections.
Well, it's still cold and moist down.
It is, yeah, and it's also very species related. So, and as everybody's probably aware of, is with the different state legislations, what you can keep in different states and that sort of stuff. So, you know, it's quite a species specific sort of thing, and that's my non-reptile vet friends that are regular vets who see dogs and cats. As vets, we joke that we're much better than medical doctors because we deal with more than one species. Well, as a reptile vet, I joke with my non-reptile vet friends that I'm better than them because I deal with even more species than what they do. And so, but it does. And the thing is, it's very different. As you'll get, you know, there's you've got snakes, you've got lizards, you've got crocs, we haven't spoken about amphibians. There's
very
all that.
Broad knowledge.
But then you get into, say, something lizards. Then you get down to skinks and dragons. Then you go down even further, and what happens in one skink species may not necessarily happen in another skink species, and so you get
water skinks are very different from fence skinks.
Correct. Yeah, you get very specific conditions that happen across those, and that's just an experience thing. Over the years you see these cases, and you know that you're dealing with something like that, whereas you will never see that in another species, and so. There's a lot of base knowledge that you build up over time.
And there's species I'll see for the first time.
Yeah.
And then I'll say.
But we don't say that though.
Of course I've seen this.
Yeah. Maybe I shouldn't.
Yeah.
But I do.
Do you find that with the geckos? I get geckos coming in. I go, I have honestly no idea what this is. And
I did a house call recently, and I was so excited because there was a number of species I'd never seen before, and I got to examine them and all that. And you know, there's basic principles that you kind of go through all of those, but different ways to handle as well, and some you just don't want to handle. Yeah. Some you're just you go, I'm going to handle this, and something bad's going to happen, or it's going to jump off and run away.
Yeah.
You know, the monitors. Some of the monitors. There's some one-offs that you see these days that they're very very rare. So, and I'm straightforward. I'll say, I've not seen this species before. Obviously, I've seen a lot of monitors.
Can I cuddle it?
Can I cuddle it? Yeah, sure you can. And but I know where to go to find out. Now the husbandry side of it, though, sometimes they'll give me a lot of information and I will learn from keepers, because if they've been keeping, I had a black-headed monitor the other day, 30 years old, that I went, Tristis, and I go,
that's an old Tristis.
It's a beautiful, and it's got a bit of arthritis, and we're actually going to do acupuncture on it, and it's actually going really well at the moment. But I had not seen a 30-year-old one before.
Yeah, that's
so they must have been doing something.
And I wanted to know, what have you been doing to get this? Because when I'm giving advice on another one or a young one, I want to, and some of that is very anecdotal, but that's the best you're going to get. Because nobody's doing research. That is
some of it's going to be genetics, some of it's just going to be. But
I
mean, pure luck. But if they are doing something, I keep it in a box
under my bed, but it's 30 years. No, that's not what happened. He had very good, very good husbandry. But you know, that's part of it. Is that you have to learn from. Some of these keepers who've been keeping for longer than I've been alive.
That's right. And an important point is not to discount those people. You know,
absolutely not.
I don't breed. I keep quite a number of reptiles. I don't breed them.
No.
Yeah.
If I need advice for someone on how to breed a particular species, I've got people. I'll say contact this person.
Yeah.
You know, because I don't certainly sit here and say I know everything about it, because there are people out there that are a hell of a lot more experienced in certain fields. But from medically,
I know I have to spay that one.
Yeah.
Well,
this one is eventually going to get this.
That is part of it. But you surround yourself with people who know more than you, and you'll do well.
Yeah, makes you look good.
Excellent. Look, as far as quarantine and testing, do you guys have any solutions for keepers out there?
If somebody has that, give me a call.
Get a pet rock.
327.
There are probably viruses that infect rocks.
That live in rocks,
What are your solutions for if you get equipment from another keeper? How can you clean it?
It is cleanable.
Bleach and F10.
F10 for a certain amount of time. Once you get rid of all
vinegar.
organic,
vinegar is next to useless. Vinegar is what you put on chips.
Yep. Look, I always thought it was weird, but heaps of people say they clean their enclosures with vinegar.
Yeah, vinegar is a good glass cleaner.
I think those people are trying to mask the smell.
Vinegar and water with newspaper cleans glass.
Yeah, it's a very good glass cleaner.
Everybody's grandma knows that.
It doesn't kill viruses. And if that doesn't work, Windex kills it.
So bleach.
But to answer that question though, I would look at it and go, firstly, what species has that equipment been used for? So if I had a bearded dragon, to be honest, I would probably not use that, because you've got an adenovirus that
18 months.
Yeah, can last years. Even if I had something as serious as something like sunshine virus, the reality is by giving that an enclosure out, a good clean out with an F10 or whatever, with whatever suitable disinfectant that you want to use, leaving that tank empty for a period of time of at least probably two weeks to whatever, keeping the tank really hot, it's going to kill those viruses.
As long as there's no organic matter.
That's right. As long as you've got a good clean out, there's no big pile of poo left in the corner or anything like that. So
right, even residue will still house the bacteria and they'll still be in there?
Yeah, you want to clean out all the organic material.
Of course, that's I mean, it makes natural sense.
Yeah, so, but this scorched earth policy where people have gone out and they've had virus in their collections and then just gone out and burnt everything, I think that's overkill. Because I don't think that's necessary.
Maybe they're just trying to keep warm in Victoria.
Well, maybe, yeah. Well, electricity costs a bloody fortune. So,
trash can fires full of reptile equipment in the living room.
Yeah.
That doesn't sound great.
Yeah.
Well, it's definitely all the burning plastic is going to be not healthy for someone. Look, as far as hygiene and quarantine, pretty much you say you don't really bother with the quarantine.
Yeah, so I get asked, one of the most common questions I get asked, and Josh probably gets asked as well, is how long do I quarantine a snake? How long do I quarantine whatever reptile it's going to be? My stock standard answer now is...
if you're going to put it in the collection, it's going to take whatever it has with it.
I do a risk assessment of what that is. If I get... Mum comes in with a son who's got his first snake and they've got a children's python, there is absolutely... what are you quarantining it for? There's nothing to quarantine.
Yeah, they have nothing else in their
collection. Do they need to test that animal? Probably not. There's the risk, what's it going to risk? Versus somebody that comes in that's just bought a new $5,000, whatever the latest morph colour in carpet python is.
And it is their breeder female.
Their breeder female, and they're about to put it in with their collection that they've been building up over the last 40 years.
Sure.
And they want to argue about the price of the virus test. That's a whole different conversation.
Yes, it is.
And so we're all nodding going, but that's a whole different conversation. And so... but like I said before, you can't out-quarantine these viruses, and so I say to people,
not with certainty.
Not with certainty. If you want to be as certain as we can be with what we know now, you need to test your animals before putting them into your general collection. So keeping those animals isolated from the other animals, testing,
good hygiene,
preventing transfer across, test them. Then if they're negative, then you're fine.
So that's for large keepers, pretty much. What about for smaller collections?
Well, if you get small collections where all of a sudden they'll have 3 or 4, and they go, "Oh, I went to somebody's place and I wanted that one, and I took 3 home with me."
Yeah.
That's a recipe for disaster.
It is,
in many cases, because.
So you would recommend getting them all tested?
If no, my recommendation is if you're bringing animals in, either permanently quarantine, and if you're getting 3, you can't, because you need 3 separate houses to do it properly, and so therefore, what are you going to do?
I look at it too. Like, I'm probably a good example of that. I own probably about 8 or 9 snakes. None of them are spectacular. They're rescues. They're nice snakes, but they're nothing.
You've met Virgil. He's got an overbutt like this.
Yeah. So they're nothing. And I might get a snake surrendered to me for whatever reason. Do I go and test it? I don't. And I can get it done at pretty much no cost. Do I get it tested? I probably should, but I don't, because I go, well, my risk, what's the worst thing that can happen from my end is I lose my snakes. Sounds awful, but I get offered snakes 3 or 4 times a week.
And you're already doing them a favour by bringing them in.
Correct.
Rather than letting them go.
Rather than letting them die.
Yeah. So I don't test my snakes for that situation, but my situation is very different to...
Your finances aren't tied up in those 8
Correct,
snakes.
that's right. It's not my livelihood, it's not my...
passion.
Well, it's my passion, but I have them because I have them, and I think it's really important
to have them. Yeah, right.
I can't see how you can be a reptile vet and not actually own reptiles, but I don't do that. But so that's a conversation that we have at the time about that, and that's hopefully with my talk and the knowledge that's getting out and this chat today, is that more and more people start thinking about it before they go and get their reptile. They go, okay, how am I going to, or what do I need to do to minimise my risk? And do it that way.
So in large collections, do you
look, a large collection.
Environmental testing as well?
Well, Josh is the man on that because he's done that.
I've done a number of them, and I think because of depends on what you're testing for.
So environmental testing just means, instead of just testing the animals, we're looking at the substrate, we're looking at equipment, we're looking at possibly drains and taps inside the enclosure. I think it is a large...
I think it's an interesting exercise.
Right.
I don't think it changes much.
Okay.
Because, again, my problem with it is if you haven't found it in any of the animals, and you do find it outside, that it means that maybe something does have it in there, or somebody's come in that has it. We just don't know. We also don't know if it's an active virus. So finding it there doesn't mean that obviously you're going to even have infectious material in your collection. So I have done it for people, and it's not something I highly recommend, but they have tested so many of their animals, got negative. They just wanted to show that. I tested outside of enclosure. I tested vents. I've tested the doors. I've tested the water supply. It was purely an exercise in showing that, right, if we did happen to come up with something, there was nothing in there at that point. If there had, we would have had problems. We would have had to start looking and investigating a bit further.
Right.
But it was that there was no genetic material for that virus, and it was only one virus. Because if you want to do that for all of the different viruses,
the amount of PCR screens.
That's how it gets to go to Hawaii on holidays.
Yeah. Well, my markup on my test is like a 0.
Yeah,
we make very little money. So I mean,
contrary to popular belief, we make as vets very little money off testing.
I'm probably very low, especially with some of my bigger collections I'm testing, and I do that because I'm also doing a lot of other health assessment there, and I'm trying to make it doable for them in these cases.
But I imagine you also want to encourage the testing.
You do, but it is something that it's a tough one, because you're spending the time, you're taking the test, you're sending the test, and it's got to be, FedEx or Express posted over there. There's a lot that goes into it. Swabbing is the easy bit. Then you've got to get it, then you've got to interpret it, and then you've got to give information on what to do with that information. And I find that's actually where the cost should come in. The cost comes in because you are giving a recommendation there. The test itself is actually, it's not that hard. You do this, and that's it. So I think that when I'm quoting a price of a test, it's not just the price of my test, it's the price of the conversation around that test, and then what we're going to do to help prevent that from spreading in the collection.
And I actually talk about that in my talk, is that I encourage reptile owners not, and this is in no way derogatory to other vets, is that I'll get people who ring me and want an estimate, a quote for what a test costs, and I'll give it to them. I go, I can get it cheaper done with my local vet. And I had a situation not that long ago where the local vet had done the test had come back positive, and then that local vet had said to this owner, just give me a call and I'll tell him what it means. And I just said, no. You want my advice, you can come and actually pay for my advice.
Yeah, absolutely.
And you should have come and got the test done from me.
That's the fair thing to do.
Correct, yeah.
It's no small thing you're doing. You're doing oftentimes, whether it's immunohistology or reverse transcription PCR screens for respiratory viruses, there's a lot of work.
But it's just that it's the interpretation of the test.
But you're giving medical advice.
Correct.
Why should you be paid for that?
And whether it's a positive or negative, there's just as much discussion that goes into a negative test as there is a positive test.
Absolutely.
Oh, I dread positives.
Yeah.
It is the worst thing you get, because you go, now you've got to tell somebody, and then you've got to figure out what to do, especially when they've got 30 or 40 animals.
Yeah, because you've just come to the realization this is going to cost them, if they want to test all these animals, you're looking at...
Well, I do look at that, and I go, who has 10 grand lying around to do a test?
And then the other worry is are they going to bring all those snakes into your clinic to test?
No.
So you've now got a waiting room full of 20 odd positive snakes.
And I know you don't do it, but that's why I do house calls.
Yeah.
For bigger collections.
Right, you don't want to bring all of those in.
I go there, minimize stress for them, and when you have big collections, I mean, you'll go out for big collections.
Yeah, I'll go for big collections,
But and I find it minimizes the risks for the clinic, for the animals, for the owners, and then...
and you just have 10 shower before you go back to work.
Yeah.
Oh, no. When I do that, no, it's a one-off day. I'm out there all day.
Okay, done for the day.
Pretty much.
Yeah, so Josh does more of that than I do. I tend to make up test kits and give them to the clients and show them how to do it, how to take the samples. You go home, take the samples, and then bring back to me.
Right. Well, both good ways to avoid bringing it into
I don't want it. I don't want it. Yeah.
Guys, I think we're going to move on to new research. Cool. We've already been here for ages. We're flying along. All right, guys, onto our new research segment. We're still talking snake viruses. So, our first paper today, Piprick et al. From 2017. First recorded case of Bornavirus infection into a healthy snake collection in Croatia. This is from BMC Veterinary Research. It was actually a virus as well. So this is Bornavirus, a negative sensed single-stranded RNA virus. This includes the virus genus, isolated from worldwide zoos and private collections. That includes colubrids, elapids, vipers, crowletid vipers, boids, so boas and pythons. So in this case study, a new Dendroaspis Jameson's mamba, anyway, in 2014 was brought into a collection in Croatia, showing respiratory symptoms for about 1.5 months, and died after 10 days of Marbacil antibiotic treatment to try to control those symptoms. Following that, the symptoms spread throughout the collection with a high mortality rate. Autopsies showed fibrotic pneumonia, and samples were collected from lung tissue, as well as tracheal and cloacal swabs for tissue culture. Paramyxovirus viral DNA purification was done using the iPrep total RNA kit, amplifying the paramyxovirus L gene fragments using one-step reverse transcription PCR and nested PCR. So they took those PCR products and then PCR'd again. That's a nested PCR. Reverse transcription PCR, by the way, is when you're transcribing RNA. So you just got to pretty much reverse transcribe it into coding DNA, and off you go, PCR away.
Makes it sound so easy.
It is, yeah, it's very simple. So it was aligned in clustal, so sequences were aligned in clustal. Phylogenetic analysis performed in MEGA, along with some prototype paramyxoviruses and similar sequences that they got from NCBI through BLAST. That's the basic local alignment search tool. On to results and discussion. Interesting, the phylogeny showed that all group within the group B of the Bornavirus genus. Now that included the Crolitis species, Bitis species, Dendroaspis species, Morelia viridis, so that's the Australian green tree python, and Aspidites melanocephalus, the wonderful Australian black-headed python, which we have just over there. So all Croatian paramyxovirus isolates showed high nucleotide identity with Germany and Austria, suggesting two possible sources. The Dendroaspis jamesoni from Czech Republic in 2014, which then spread to their Crolitis molossus, of course being an identical strain. Now the second source was a possible green mamba, Dendroaspis viridis, and a Bitis nasicornis. What is nasicornis? Is that a horned viper? I didn't.
No idea.
Check that one. In 2015.
We don't have them here in Australia.
No, we don't. Anyway, these were the source for the strain, for the second strain. The source for the C strain in Crolitis molossus pyrrhus was unknown and appears to have been in their colony since 2013. So, it appears that these new viruses came through from new snakes not previously tested or showing any signs of disease, as we've been pretty much talking about. Since this is directly from the discussion there from the authors. Since no vaccine is presently commercially available, basic veterinary health checks and quarantine measures are of great importance to minimize the risks of infection. Your thoughts? Start with you, Ahlia. What do you reckon? This is pretty shocking to see just either one or two snakes come in and wipe, and you know, this includes Australian species here as well. So this has got something that they've jumped from a mamba to their tree pythons and their black-headed pythons.
Yeah, I was surprised when you said that.
Yeah, Australian species in there as well, right?
Was it like very similar symptoms in all of them?
I believe so. From memory, it was all very similar. Respiratory symptoms. Not much more than that. Just a lot of mucus, and did they have stomatitis as well? I believe they had some swelling.
Most likely they probably did.
Yeah.
Given
anytime inflammation in the respiratory tract generally.
Generally these stomatitis, I imagine. But, So I imagine the symptoms were pretty similar.
Yeah, and is that pretty unusual to have very similar symptoms in such different species?
And not only just different species, but different classes of
different
reptiles.
classes.
I find that
lapids, colubrids.
Sounds pretty dangerous then.
Yeah.
Something we don't want getting
unusual
out.
for a paramyxovirus.
Yeah.
To be so widely spread. Yeah. Right. They're usually quite
well,
I mean, it is Furlovirus
so far the
as
ones
well.
I've been looking at, but
well, Furlovirus is named for a species. It's named for the fur to land.
Which is a whole interesting story because they actually got that wrong, I think.
Oh, really?
Yeah. Are you going to tell that story tonight?
I'm going to tell that story tonight.
Oh,
so it's actually not from fur to
Sneak
lance. They think it might. Well, it's now confused, but there was possibly Brazilian lanceheads was the actual original snakes that were infected.
Okay.
So, but the other thing too about Furlovirus and something people have probably heard of OPMV. So
Ophidian paramyxovirus.
Yeah, so that's the layman's term, if you like, of what Furlovirus was called. And that term was never has never been officially recognised.
No.
So
right.
And so whenever you hear people talk about OPMV, they're actually what we should be talking about is filovirus. And what's also important in this little study or paper here is that there's at least 4 subtypes of filovirus, and they're finding different subtypes in these
and
animals.
Able to track them around.
Correct. Yeah. So, and they may end up being completely 4 different species of filovirus, but there's different types of filovirus is what they're being found. So, but that's scary. You know, we were talking earlier about whole collections being wiped out. There's an example.
There you go, right there in Croatia.
Talking about what's going on up north, and that is jumping major classes.
Yeah. Well, it's across lizards, turtles, crocodiles, fish.
Yeah.
Well, Bornavirus. It's
Bornavirus is an interesting one because it's I don't know what percentage of as humans our DNA is actually Bornavirus. Right. So we've all got Bornavirus in us. Yeah,
so because of the crossing back into the human genome. Yeah.
The most is usually in birds with their ventriculus.
Yes. Yeah, we're not bird vets, so. For a good reason.
Right.
So, but it's an interesting paper, that one.
Yeah, a nice short case study there, but definitely showing some of the risks.
Yeah.
All right, moving on. Keller et al. 2017. Co-infecting Reptorinoviruses can be vertically transmitted in boa constrictor. So vertically transmission, guys.
Yeah. I think there's that one case of sunshine virus.
So vertical transmission meaning
mum to baby.
So mum to baby.
Wow.
So horizontal, individual.
Horizontal is from animal to animal.
So to
this
offspring.
Is mum to baby. Yeah. So or dad to baby. But,
and of course, Reptorinovirus, as we were talking about, the likely cause of boa inclusion body disease, recognized since the 1970s, a transmissible, often fatal disease with variable symptoms, often respiratory and neurological. So again, hard to untie from the rest
Everything
of all those
else.
diseases.
Yep.
The one thing that does kind of give it away is those inclusion bodies, that it's the name.
Others get inclusion
but
bodies.
Again, I think we get into that in this paper as well. Not all of them show inclusion bodies.
Or not only this one has inclusion bodies.
Not all inclusion bodies are caused by Reptorinovirus. Is that correct?
That is correct.
So transmission routes and incubation period up until this time were unknown. Proposed vector was direct contact or via snake bite. With Reptorinovirus found active in arthropod cell lines, so there is a possibility there. However, there's few studies on vertical transmission in reptiles. That said, it has been seen in adenovirus, herpesvirus, and recently in sunshine virus. So the question here was, can Reptorinovirus be vertically transmitted? And they examined 5 boa constrictor clutches from 2 breeders in Switzerland with confirmed Reptorinovirus infection by traditional histology and immunohistology. And of course confirmed a lot of the time throughout this study with reverse transcription PCR in parents diagnosed with boa inclusion body disease and in offspring from those parents. So first trimester offspring all the way through to 20-month-old juveniles. This was the PCRs, histology, immunohistology, all done on blood smears or post-mortem tissues from brain, heart, lung, liver, pancreas, and more for establishment of cell cultures and or RNA extraction and next generation sequencing methods to ID multiple Reptorinoviruses in a clutch. They would then confirm any next generation sequence hits with reverse transcription PCR using very specific virus-specific primers from previous studies and of course from their de novo genome build with the next generation study. They built some maximum likelihood trees with the de novo assembled viral L and S genome segments. Very, very cool. Now, results in discussion really shows some of the impacts with those poor clutches. Horrible clutches. Perinatal abortions. All kinds of terrible health issues and a whole lot of euthanasias before they even got very far along.
We find that with a lot of virus stuff that you get abortion related. That's in what's the one in people at the moment? That's what's
right.
Zika virus.
Thank you.
Zika virus, right? Which is obviously vertically transmitted and then causes very similar health issues.
It does seem to be viruses do a lot of that because if it's happening in the developmental portion of it, they don't develop right.
Right, now as we were saying as well, directly from the authors, it was overall surprising to see how many offspring exhibited Reptorinovirus infection without evidence of inclusion bodies or inclusion body formation or viral antigen expression. So you can very easily, so for example, Clutch 4 had no signs in cytology or histology of inclusion bodies, but post-mortem revealed, of the father revealed inclusion body formation and expression of the viral antigen, and the offspring were reverse transcription PCR positive for the virus. So they had it, they didn't have the inclusion bodies, but dad did. So the inclusion bodies maybe take a little bit more time to show up?
I think it's a virus affected by what type of virus? I think it affected what species you're dealing with and what age.
Right. Okay.
Yeah, and
so both species and age.
Yeah, and so I think the reality is that finding inclusion bodies is not really a diagnostic. It doesn't tell you what specific virus you're dealing with.
Right, it's just a symptom that happens at some point.
And the thing is, other things other than viral infections can cause inclusion bodies.
Okay.
So,
right.
Yeah.
So that, I guess, immunohistology check for it is probably a little bit less reliable than, as is often the case, I guess, for viruses.
Yeah, I think PCR is gold standard these days. You need it for diagnostic because otherwise you're going to miss it. And even then, if it's not shedding it, unless you do histology on whatever target organ it is, you're not going to find it.
Yeah, right. So directly here from the paper as well, the results indicate that it can take several months before a prenatally infected snake exhibits definite signs of BIBD. That's boa inclusion body disease, of course. In any case, our results demonstrate that animals with boa inclusion body disease / Reptarina virus infection should not breed, since the likelihood of offspring becoming infected is high. So it looks like they pretty directly transmit that vertically to pretty much all of their offspring. Is that often the case with a lot of these viruses, do you think? Or
I haven't done enough testing to. I don't have enough numbers for me to say about my own.
So there's one published thing in Sunshine Virus about. Actually, I'm not sure it's published. I think it was just presented at a.
They did say that there was published vertical transmission.
Diamond python.
No, there's a coastal carpet pythons in Sydney that were. Mum and dad were positive, infected, had a clutch. They tested the eggs. They found the fetuses, the embryos, and the membranes inside the eggs carried the virus.
Membrane as well. That's right.
That's right.
I do remember reading them.
But some of those snakes hatched, and those snakes have since tested negative.
Negative. Yeah.
The ones that actually
hatched
all had
positive.
Okay. And I've had exactly the same situation in 2 collections.
That's kind of handy.
I actually haven't done those tests yet.
That's kind of handy that the affected eggs fail to.
Correct.
But have they just not showed up yet because they're saying that. Did they test positive from the beginning? Sorry.
Yes.
No. It takes several months.
So it takes several months.
Yeah.
So they've got to continue. They need to test those snakes again
over their lifetime.
So
that's interesting. Well, hopefully, Reptarina virus.
I think the moral of the story is I wouldn't be recommending breeding positive snakes.
No.
Any of the viruses to do that.
No, if you're getting positive viral PCR, probably give it a miss.
Breeding's probably not the thing you need to concentrate on.
Antivirals. I think human doctors will say the same thing. Hoon-Hanks et al. 2018, respiratory disease in ball pythons, Python regius, experimentally infected with ball python nidovirus. This is from the Journal of Virology. So, bit of a rough paper here, but such is science. So, order Nidovirales. A large, diverse group of viruses with known human and animal pathogens recently described a new group. They didn't recently describe, but there is a recently described new group which infects lizards, snakes, cattle, and even nematodes, small worms. So again, we got very, very broad virulence and infection. It has been proposed reptile nidoviruses be classified in the distinct Bornavirus genus. For example, ball python nidovirus to be classified as a Bornavirus, hopefully. Well, that's what they're suggesting. Originally discovered in ball and rock pythons, that's Python regius and Python maloris. It presents as a respiratory disease again, with many symptoms: mucus, stomatitis, proliferative pneumonia, and a lot of secondary infections. Is that kind of due to the lowered immune system, or just the mucus thing? Do you guys have a...
I think anytime you break down the mucosal barrier, you're going to predispose to increase or any lower immune system. You're going to predispose to bacteria which already live there. They're all commensals. Most of these bacteria that we find are stuff that lives in them, which is why you can't just swab them out and test that, because it doesn't mean anything, because those bacteria could just be living there.
Right.
So you need to kind of tease out which bacteria is causing the problem.
So they're already present in the animal,
Correct.
And then the mucus...
It's like a skin infection. You get a staph infection. Well, staph lives on your skin.
It's there. Your immune system copes with it. Something has to cause damage for it to become pathogenic.
And viruses are very good at that. We see viruses causing primary disease.
And Josh and I will see that all the time. We'll get second opinions on cases that the vets have all they've done is swab them out.
Right.
And then they've cultured some growth of bacteria in there and put it on an antibiotic. And the reality is that
they
culture,
don't know
a complete
if
waste
they've
of,
cultured, they usually just look on the microscope.
it's a complete waste of time. So.
Well, either way, it has been detected in oral swabs in Burmese bull, Indian rock, and green tree pythons, carpet pythons. So again, Australian species that we are both very well aware of. Boa constrictors with and without respiratory signs. You can see all that in another paper, Marchang and Kolesnik, 2017. Now, the aim of this paper was through experimental infection to conclusively establish a causative relationship to respiratory disease, characterize the clinical course, explore useful diagnostic techniques, and to investigate transmission routes. On to methods. Let me take a breath. They took Borrelia spilota heart tissue and created a cell line, euthanized for its spinal issues. They took oral swabs from a Python regius colony with ball python nitivirus. 1 ml of the swab extracts were applied to the python tissue cultures created from that heart tissue, and they monitored the cells for cytopathic effects daily. 7 days post-infection, the cells were frozen at minus 70 degrees, obviously killing any living cells, but leaving the viral DNA. This was then thawed and used to re-inoculate new cultures, from which they did plaque assays, where you basically count the number of degenerative growths to kind of get a rate of infection. Next, they took 5 6-week-old Python regius, so that's ball pythons. The 3 termed, which were called A, B, and C, were infected orally and intratracheally with the viral culture. D and E were kept as controls. Python C was euthanized early at 5 weeks on after starting to show symptoms. A and B were allowed to keep showing symptoms well on up to 10 weeks and euthanized. D and E were both euthanized at 12 weeks. Now, pre and post-infection, choanal, so that's a nostril area, oroesophageal and cloacal swabs were taken. RNA from swabs, as well as from frozen tissues from the lung, trachea, liver, and more, were used for RNA extraction, followed by quantitative reverse transcription PCR, which is a bit more complicated than regular reverse transcription PCR. Basically, between rounds, you use photospectrometry to assess the PCR product production, which is kind of a proxy for gene expression. So this was done using nitovirus primers. They also did metagenomic sequencing by shotgun library, where you basically break down the whole genetic material that you have in there into little chunks, sequence it all, and then they filtered out the Python-specific sequences, leaving only the viral sequences, which were blasted, which is, again, the basic local alignment sequence tool on NCBI, against other viral... they blasted that viral DNA to find other similar sequences. Now, antibodies from GenBank for the black-headed python and, for the ball python and the green python nitovirus nucleocapsid proteins were collected and analyzed and then submitted for synthesis, and polyclonal anti-nitoviral nucleocapsid anti-sera were developed in 2 rabbits. Histopathology and immunohistochemistry was done on the tissue samples. Cultures were also analysed by Western blot for viral gene expression. All right, there we go. That's a lot of methods. On to the results and discussion. So clinical signs, 4 weeks post-infection. That's pretty quick, right? Is that what you pretty much expect for...
That's what's being seen in ball pythons.
Yeah, okay. They're just on like that.
Yep.
Okay. With progressive oral reddening and mucosal secretions and hemorrhages, leading to increasing respiratory effort, open mouth breathing, anorexia by 10 to 12 weeks, confirmed infection by cytology and histopathology. The open mouth breathing, that must be getting pretty severe there, because, correct me if I'm wrong, pythons have pretty large lungs. To get them to struggle to breathe, they must really be...
It's the trachea, that's the issue.
It's the trachea.
It's very narrow.
Okay. So once it gets inflamed...
Inflamed and secretions, effectively choking.
Yeah, okay. So they're choking on their own trachea and the secretions in it. Mm-hmm. Wow, that sounds like fun.
And they can't clear it because they can't cough it up.
Oh, they can't. Yeah, of course they can't.
I don't understand the use of the word anorexia.
So, just not eating.
Yep.
Yeah.
But I feel
losing large amounts of weight. Maybe not used in the same way it's clinically used for humans.
Yeah, that's what
So anorexia in humans is a psychological disease, whereas anorexia in our world is not eating.
Okay.
Right. So anorexia by 10 to 12 weeks just means
not eating.
Well, correction, people can be anorexia. You can have anorexia but not have anorexia nervosa, which is the actual...
okay, yep.
Anorexia nervosa is the
psychological
behavioural component. Okay. All right, so antemortem swabs and postmortem tissues tested positive for viral RNA and, of course, proteins. And infectious virus was detected, of course, by PCR very frequently. Also detected by the immunohistochemistry, and they detected viral RNA expression in Western blot, and they found the virus in isolation in the plaque assays. So quite easy to find ball python nidovirus once you experimentally transinfect them. Our findings demonstrate that BPNV infection caused marked mucinous inflammation of the URT and cranial esophagus, with progression towards proliferative interstitial pneumonia. So the virus is released in respiratory and oral secretions, and fecal transmission also appears possible. Is that pretty much what we're seeing for nidovirus? Oh, nidovirus, remind me, are we seeing much of this in Australia?
Yes, so we have a nidovirus, but not this one.
A different
right,
one.
yeah, we have the shingleback.
And a python one.
And that's what's interesting in this paper is they refer for the first time here to this Bornavirus genus.
Right. We were referring to it as coronavirus.
Yeah, so when it was first described here in Australia, it was called coronavirus because nidovirus is the
that's
bigger group.
New genus that they're talking about.
Yeah, well, it's the order of the order is nidovirus, and then underneath that was coronavirus. Well, now we are referring to it as nidovirus.
Recently been told no, we have to go back to calling it nidovirus.
But this Bornavirus, so what they've done now is they've created this new group, and all of these reptile nidoviruses, so shingleback nidovirus 1, the nidovirus that we have in our python here, they talk about ball python nidovirus, Marili viridis nidovirus, there's a rock python nidovirus that they call python nidovirus, are all going to be included under Bornavirus.
Within the Nidoviridae.
Okay.
Correct, yeah. So now, what's also interesting, because I like to know the names of how they get these names. So sunshine virus is named after the sunshine coast. Virus is named after the Fertile virus. Bornavirus actually stands for, Josh, do you know what this means?
Nope.
No, I had to look this up. I actually contacted Tim and said, where did this come from? It stands for bacillus, is the BA, because it's got something to do with the structure of the virus. R is for reptile, NI is for nidovirus, and the virus is the virus. And the naming
Bacillus
actually comes
reptile nidovirus.
Correct. There's actually a bacillus fish nidovirus that's called Banfiviruses.
That's actually where the name.
I've
heard of Banfiviruses.
So there you go, Josh. I've taught you something today.
Love it.
That's where Bornavirus
That's a good way to
come
name
from.
things.
That is fantastic.
So there's a group in the nidovirus called Banfiviruses,
and then
I
there's
was
also
wondering who the guy named Barney was.
I'm thinking, I don't...
Sunshine was found in the sunshine. Was Bornavirus found in the barn?
I thought that all viruses now are named after the location you find them, and I'm thinking, I've never heard of Barney. Like, where the hell is that?
Any
that's
barn,
actually where it comes from.
whatsoever.
It's the structure of the virus from the species of the virus or the type of virus.
Fascinating.
They call it
Good stuff. Love it.
But the other thing that's important about this paper is this is the paper that actually definitively says that ball python nidovirus is the cause of ball python nidovirus.
This was the first one where they really got the transmission route absolutely down.
Yeah, and proved it. So, but this is a problem. Ball python.
Evans postulate?
Cox postulate. Cox postulate or Evans? I don't know. One of them. some scientists will be able to tell us. But yeah, this is ball python nidovirus is a big issue in the US.
Right. That was a chunky paper. If anybody wants to read that one, that's a solid one. Who hanks it all?
I'll read it
That's a heavy one, boy.
Reading that one.
You can read it to me on the way home.
All right. Guys, I think that's about it for the show today. Thank you so much, everybody, for joining us. Ahlia Chanel, a joy as always.
Thank you.
And we've got, of course, Dr. Joshua Llinas. Has been a pleasure, and I'm sure we'll see you on here again soon.
For sure.
Dr. Shane Simpson, thank you so much for joining us. Are you up here frequently? Or?
Well, I do get up here probably to Brisbane at least once or twice a year.
Cool.
Having lived up here and gone to uni up here.
Sure.
But up for RepX this one, and I'll be back up in May for the Australian Veterinary Conferences here in May, and I'm giving a lecture at that. So
excellent. Well, look, if you're ever up in the area and you're keen to join us again for some more cake and mayhem and cocktails, we'll definitely see you at RepX tomorrow. Everybody check that one out, repx.com.au. Now, for you guys as well, you can check out HerpVet at herpvet.com.au or on Facebook, and the website is live as of tonight.
Woo! Can't wait.
Excellent. And what are we doing to celebrate? We haven't...
I already did.
He's coming to my talk to listen.
More viruses.
More viruses.
And then go to the Red Room.
That's what we do.
And you guys can follow Shane at thereptiledoctor.com.au. You can also find him there on Facebook. Where else can they find you? Are you?
I'm everywhere. I'm like a rash.
Yeah.
I'm like the new reptile virus.
The new reptile virus, nidovirus.
Absolutely.
All right, wonderful. All right, thanks so much, guys, and plenty more wildlife cake and cocktails coming up in the next couple of weeks. I'm Yanni Torcola, your host. We'll see you soon. Cheers, guys. Bye.